Have you ever wondered how insulin is formed in a laboratory? It's a fascinating process that involves a blend of biology, chemistry, and a bit of scientific wizardry. So, sit back and relax as we delve into the intricate world of insulin production. Firstly, it's crucial to understand what insulin is. This hormone, produced naturally by the pancreas, regulates the amount of glucose in the blood. For those with diabetes, their body either doesn't produce enough insulin or doesn't use it effectively. That's where lab-produced insulin comes in, a true lifesaver for millions worldwide. The journey of lab-produced insulin begins with the humble bacterium Escherichia coli, more commonly known as E. coli. Scientists introduce a small circular piece of DNA known as a plasmid into these bacteria. This plasmid carries the human insulin gene, essentially instructing the bacteria to produce insulin. Now you might be wondering, how does this tiny piece of DNA get inside the bacteria? Well, it's through a process called transformation. The bacteria are made competent or able to take up the DNA by treating them with a solution of calcium chloride. This alters the bacteria's cell membrane, allowing the plasmid to enter. Once inside, the plasmid makes itself at home. It integrates into the bacterium's own DNA and begins to multiply as the bacterium divides and grows. As the bacteria multiply, so does the insulin gene, leading to the production of insulin. But we're not done yet. The insulin produced in this way is actually proinsulin, an inactive form of the hormone. To convert this into active insulin, scientists use an enzyme to cleave the proinsulin, creating the final product. The insulin is then extracted from the bacteria, purified, and finally, it's ready for use. It can be injected into patients who need it, helping to regulate their blood glucose levels and keep them healthy. In summary, the process of insulin formation in a lab is a multi-step process that involves the use of bacteria, DNA manipulation, and enzymatic reactions. It's a process that showcases the power of modern biotechnology, allowing us to produce life-saving medicine on a large scale. The importance of this process cannot be overstated. It's not just a fascinating scientific endeavor. It's a vital part of managing and treating diabetes. Without this lab-produced insulin, millions of people worldwide would struggle to control their blood glucose levels, leading to serious health complications. So the next time you ponder the marvels of modern medicine, remember the intricate process of insulin production. It's a testament to human ingenuity, a shining example of how science can truly make a difference in people's lives.